So we just finished the chapter 1, DNA, RNA, and now the chapter 2, we will discuss nucleic acid-based cellular activity. So basically, not only chapter 2, but also chapter 3 and 4, we will discuss all these cellular activity that involve the nucleic acids, right? So those activity including, for example, here, the DNA, right? Perform the expression, transcription, translation, cell growth. So this is DNA-based activity. And then the replication, right? DNA replication, that's also a DNA-based activity, cellular activity, and also the horizontal transfer, that's also DNA-based activity. And this chapter, we will focus on the replication, DNA replication and DNA repair. And because this chapter is a huge chapter, so basically we will just finish half of the chapter 2 today, okay? So the DNA replication hypothesis. And the DNA hypothesis, the replication hypothesis in the early days, all right, there are 3 different proposals. So they believe that DNA could be replicated by the 3 different mechanisms. One is called the semi-conservative mechanism, and semi-conservative mechanism, that means the DNA replication, in the end of replication, each daughter strand, each DNA, they will get one old strand and one new strand. And then for conservative, basically, after the replication, O1 will combine with O1, new one will be new one. Disperse, basically, just disperse is randomly replicate. Okay, so that's called disperse. In order to prove what is the correct mechanism for the DNA replication, so these 2 scientists, they perform this experiment, okay, to confirm that the DNA replication is semi-conservative. Okay, it's a Matheson and Starr experiment. They basically they grow the bacteria, okay, nitrogen 15, okay, nitrogen 15 isotope to the nitrogen 14, and then after they grow several generations, and then all the DNA in the bacteria should contain only nitrogen 15. And then they transfer the cell, right, to the nitrogen 14 media, and then they grow 1 generation. Okay, after 1 generation, they collect the cell, and then spin it down, and then analyze it after centrifugation, and analyze the location of the DNA, and then they can determine where the location of the DNA, and then also they collect the cell for 2nd generation. All right, after 2 generations nitrogen 14, so that they can also collect the cell and then perform the centrifugation and determine the location of DNA. So from this experiment, basically, the idea is after this centrifuge the DNA they will be in different location based on the weight of DNA. For nitrogen 15, because it's heavier, so therefore will end up in the bottom. If it's a hybrid, nitrogen 14, nitrogen 15, then it will be in the middle. And then it will be, if it's nitrogen 14, then it will be on the top, all right? So based on this, they can distinguish, they can determine which mechanism is the DNA replication. Okay, so I give you this preview, and now this video will give you a detailed explanation. Okay.
The Messelson and Stahl experiment provides evidence for semi-conservative replication of the DNA molecule, where the 2 parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Messelson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
Do you understand?
So the parent strand is the strand that is the one that's used to create the second strand for every generation.
Right.
Gotcha.
So for this experiment, okay, the first, the parentals, right, they have this nitrogen 15. It's heavy, all right? So if you collect the DNA, then you centrifuge, will be in the bottom. And then so to determine what kind of a mechanism for the DNA replication. So they move these bacteria to the nitrogen 14 for one generation. Then they collect the cell and harvest the DNA. And then after the centrifugation, all right? So let's say this N15, if it's conservative replication. So that means after replication, then you have a one double strand will be nitrogen 15, and then another double strand will be nitrogen 14. All right, and then the after centrifugation, then you should see two bands. One is for nitrogen 15, one for nitrogen 14. If it's a semi-conservative replication, then after the replication, you will see the mix like this. So 14, 15 hybrid, 14, 15 hybrid. So then when you perform the centrifugation, they will only show one band because all the population will be this hybrid. And that's what they see. So that's why they show the after one generation, they see one band. So it's a 14, 15 hybrid. Yes, Marina.
So by combining two types of DNA, they can see as an outcome the route of replication it takes?
Yeah, that's right. Yeah. Okay. So that's why they perform this Mahama, yeah.
My question is, how do you know?
Yes, I'm in the process.
So you have two separate kinds of DNA strand, and you combine them, like Marina said, and then that's how you end up finding out whether or not it's conservative or semi-conservative. But how do you know what is the orientation of each strand before you combine it? Like, how do you know which one is, like, how do I know if I'm putting in semi-conservative and semi-conservative, or versus like conservative and semi-conservative?
Okay, so
Yes,
when they perform the experiment, they grew the cell, and then they provide only nitrogen 15 in the media. So when they synthesize the DNA, they will only introduce nitrogen 15 in the DNA. Okay? So all the cell contain only 15. Then they shift the cell to nitrogen 14 media. So that means the cell will use the nitrogen 14 for their DNA synthesis. All right. So when they perform a replication, that means this is DNA synthesis. And then they introduce the 14. All right. So if it's conservative, like this one conservative, so this is 15. After this DNA synthesis, the replication, then you will see one population is 15, one population will be 14. If it's semi-conservative, then you will see only one population because everyone will be this 14, 15 hybrid. And so that's how you distinguish the difference.
So our DNA is semi-conservative because it has the complementary the parent and then the complement strand, correct?
Yeah, that's right. Okay, semi-conservative. Yes, very good, Marina.
That makes more sense to me. I'm sorry if it was a silly question. Thank you for.
No problem. No. Every question is a good question. Okay. All right. Very good.
Thank you.
Thank you. Question. Very good. All right. And now, so the DNA synthesis. That's the replication, right? So DNA synthesis. So very important because that will be relevant to the next chapter about transcription. For DNA synthesis, you need to have this the hydroxy group S3' so that you can add the DNA nucleotide because the DNA synthesis requires this oxygen, okay, the hydroxy group to attack the alpha phosphate group in the incoming nucleotide. So when you want to add this nucleotide here, this hydroxy group attack this alpha phosphate here. All right, don't ask me why they attack alpha. You have to ask Ogo professor, right? And this reaction is called SN2 reaction. Okay, when they attack this alpha phosphate, they will release this 2P. Okay, 2P pyrophosphate. They will be released, and then they will form this phosphodiester bond. And this is so-called SN2 reaction. And so during this process, they will release the pyrophosphate. Okay, remember this process. This process is extremely important, especially later on when we discuss the DNA sequencing, all the sequencing technology based on this principle. Okay, so this is the mechanism of DNA synthesis. Remember, you need the hydroxy group to attack the alpha phosphate, and then release the pyrophosphate and give you connect the form this phosphodiester bond. All right, and this is just the cartoon to show you the reaction. So this hydroxy group, all right, this one attack this alpha phosphate and then release pyrophosphate, and then you can connect. And the DNA replication model, right? So again, 3 different mechanisms: continuous, semi-discontinuous, discontinuous. So continuous, that means for both strands, as we know, they run anti-parallel 5 to 3, 3 to 5. And then they say continuous. That means one strand you synthesize 5 to 3, the other strand synthesize 3 to 5 continuously. But we know this is wrong because DNA synthesis always 5 to 3, all right? 5' to 3'. So this one is wrong. Discontinuous. Again, similar idea, like, so 5 to 3, 5 to 3, basically, they open up, okay, 5 to 3, 5 to 3, all right? And then they do it, they perform this segment by segment for both strands, all right? This is also wrong because it has to be semi-discontinuous. So for one strand, 5 to 3 will synthesize continuously. And then for this one, 3 to 5, they will be discontinuously. So they will do it region by region. So we have one region, 5 to 3, and then when you finish, another region, 5 to 3. Okay? And so later on, as scientists confirm All right, the DNA replication model is the semi-discontinuous model. So we have this blue one, okay, 5' to 3' be synthesized continuously. So this is so-called leading strand. And then the other one here, okay, you have to do the 5' to 3' region by region, and this one discontinuously, and each fragment, all right, it's about 1 to 2 kb long, and we call this fragment is Okazaki fragment. Here, Okazaki fragment, okay, Okazaki fragment. Remember the name, okay, very important. I remember in my last class, okay, when I had a test, I asked what kind of a fragment. Okay, it's Okazaki. And then some student give me the answer Kawasaki, Yamaha, everything here. All right, remember it's called Okazaki fragment because the scientist who discovered this fragment, this continuous replication, is Okazaki. So that's why we named it Okazaki fragment. Okay. And this is more detail about this replication in the both eukaryotic cell or prokaryotic cell. Once you open up the double strand start a replication, they have to go both way. Even the bacteria cell, they go both way. Even it's a circular DNA, right? So they go both direction. And for eukaryotic cell, it's even more important to do this because they have to identify the place where we start a replication. It's called original replication. Then you start a replication. Then because the eukaryotic cell, the DNA genome is very long, so you have to find hundreds, thousands of original replication to simultaneously start a replication from both directions, so that you can complete the replication very fast. Okay. So now let's watch this another video, DNA replication video.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So that's the DNA replication process. Okay, so remember, one strand, a leading strand, the other one is lagging strand. Lagging strand, we call it Okazaki fragment. Many Okazaki fragment. Okay, so as you can see here, many Okazaki fragment, because each region is only 1 to 2 kb long. All right, and then they have to synthesize 5 to 3, 5 to 3. In the end, they will connect, they will link each other. All right. And during that process, you can see we have several enzymes that participate. Yes, Ali.
So both times it's still 5 to 3 regardless. It's just that the
Correct.
lagging strand is the Okazaki fragments, while the leading strand is being replicated continuously.
Correct.
Got you.
Very good.
Okay.
All right. So during that video that you can see, all right, for the replication, you need to have helices to open up the double helix DNA. All right. And yes, Marina.
Are we talking about DNA replication in a prokaryote cell or in a generalized fashion?
So we use the bacteria as the model, but it's all the same in eukaryotic cell and bacteria cell. Okay.
Okay.
Similar mechanism. Okay.
And what is the difference now? Because I don't want to get confused in between the Okazaki fragments and you said another one. I don't know how to pronounce it.
Okay. Oh, no, forget about others. I was telling you that before, some student give me a different kind of name because it's Okazaki is Japanese name, Okazaki fragment for the lagging strand. But some student during the test, when I ask, they just give me any other random Japanese name like a Kawasaki, for example. It's wrong. Okay. It's Okazaki. Ozaki.
Okay.
Okazaki
Thank you.
fragment. Okay, Okazaki fragment. All right, so for Okazaki fragment, all right, that's lagging strand. The leading strand, they synthesize continuously, all right, 5 to 3. All right, and then for the leading strand, you only need one primer. Okay, one set of primer, because the primase will come to the leading strand, and then they form like a 7 to 9 nucleotide long of primer RNA. Okay, that's RNA, so that DNA polymerase will continue to synthesize the DNA. And then for the Okazaki fragment, the lagging strand, each section will require the one primer. All right, so one Okazaki, one primer, two Okazaki, two primer, and then the DNA polymerase. And for bacteria, we call DNA polymerase 3. They will synthesize the new DNA. Okay. And then, of course, when you open a double helix and the DNA, all right, they are the single-stranded, and so you need a single-stranded binding protein to stabilize the single-strand DNA. And as we discussed in chapter 1, when you open a double helix, you introduce the positive coil positive supercoil in front of the replication fork. So you need a DNA gyrase, okay, that's a topoisomerase 2, okay, in front of the replication fork to release the tension. Otherwise, the DNA will break, okay. And then after the replication, you need the exonuclease to remove the primer. And then after you remove the primer, then DNA polymerase 1 will come in to fill in the gap. And finally, DNA ligase to repair or to form this phosphodiester bond. And that's the whole process for the DNA replication. Okay, so we need a helicase to open that double helix, single-stranded binding protein SSB to stabilize the separated single-stranded DNA. And as you can see, DNA helicase, they bind at the 5' to 3' strand, which will be the lagging strand. Okay, here you will form the lagging strand, and the helicase they will run from 5 to 3. Okay, yes, Marina.
For the stabilizing protein, so when you unwind the DNA, that's suggesting it's very unstable, correct?
Yes, that's right. And also, those single-stranded DNA region is subject to all kind of the enzyme
Like
activity.
molecular reactions and everything?
Yeah, exactly.
Okay.
So that's why you need SSB to protect those single-stranded DNA region. And the helicase, okay, DNA helicase bind only the lagging strand because they have to go from 5 to 3. And this is the place where you have the Okazaki fragment.
Okay.
And then the gyrase, okay, that the topoisomerase 2 to remove the to convert the positive supercoil to negative supercoil, right? And then primase, right? Primase, that you need a primase to form this RNA. And this the function of primase because they can provide a free 3' hydroxy group to initiate the DNA synthesis. Okay, each Okazaki fragment will require one primer because we have several Okazaki fragments, and so that's why we need many RNA primer. And leading strand only one primer. Yes, Muhammad.
Is the location of the primer replaced by strands when the polymerase
So after
reaches the
the
other side?
after the DNA polymerase 3 complete the synthesis, and this section will be degraded by exonuclease. Exonuclease remove this, and then DNA polymerase 1 will come in to fill the gap.
So that's
Okay.
when the polymerase reaches the next primer, because it's going from 5 to 3'. So like there's the completed
Okay,
part,
so
like the previous Okazaki fragment is completed already, so it will reach the primer from the
no prime. Okay, let's say for example this is leading strand. All right, so you form this primer. All right, primase form the primer, 7 to 10 nucleotide. Then you have 3' hydroxy. Then DNA polymerase 3 will continue from this nucleotide and then synthesize everything. When they reach the end, okay, then exonuclease come here to remove this RNA. All right, then you have a gap here, and then DNA polymerase 1 will come to synthesize this. All right, and once they finish this, and then of course ligase repair this phosphodiester bond. Okay, so that's the leading strand. So you remove this and then fill it for the Okazaki fragment. So this one can be in the middle, all right, in the middle because this is one piece, one Okazaki fragment in the middle of the strand. Then again the same, you remove this one and then you fill in the gap and then you repair the both sides phosphodiester bond so that you can connect.
The polymerase does it goes back again for the same
Another polymerase
section.
another
Polymerase.
Another new polymerase, because the original one is called 3. Okay, DNA polymerase 3. That's the original one. And then when you remove this RNA primer, that's another DNA polymerase. It's called DNA polymerase 1.
Oh, okay.
Okay, just like this. So when you remove this, all right, And then you have these fill in. And then ligase repair these phosphodiester bond on both ends.
I see. Thank you. Thank you.
And then here, so RNA-SH, they can only cleave the bond between the two ribonucleotides. And then for the bond between the RNA and DNA, then you need the exonuclease so that you can remove this completely. All right? So RNA-SH remove most of it. And then finally exonuclease to clear up everything. And then, as I say, there are 3 different types of DNA polymerase. Okay, 1, 2, 3. 3, that's the one that perform the major reaction. Okay, to synthesize the DNA. That's a DNA polymerase 3. Okay, and then DNA polymerase 1. That's the DNA repair RNA primer excision. So we remove RNA primer. Then DNA polymerase 1 will come in to fill in the gap, or they can be used for DNA repair. And then another one, DNA polymerase 2, the actual function is not clear, but it might involve the DNA replication repair. Yes, Ali.
So I just need further clarification. So primase is the enzyme that puts the RNA primer onto the DNA strand when it's beginning to replicate.
Yes, primase synthesize the RNA nucleotide, and we call it primer.
Right, that's what causes the primer. And then the DNA polymerase 1 is the one that removes it.
No, it's the RNSH and the exonuclease remove it. And actually, yes, the exonuclease, from the DNA polymerase 1, they can remove it.
Okay, so the DNA polymerase 1 is what removes it, and then DNA polymerase 3 goes and fills in the gap.
No, 1 fill in the gap. 3, that's the original polymerase to synthesize the majority of the DNA. Okay. Here will be 3. Okay, to synthesize.
Right. So 3 is okay.
3 is the major one, and 1 is the one to fill in the gap.
Okay, so the 3 is the one that creates the strand, like that replicates the strand, and 1 is the one that fills the gap when the RNA primer has been removed.
Correct.
Got you. Okay.
Mm-hmm. All right. So, yes, Mama.
I'm sorry, just quick question to follow up. So there is 2 active polymerase 3s, one on the lagging strand and one on the leading strand, like simultaneously?
Yes. Simultaneously, yes.
Okay.
Yes.
Okay, thank you.
All right, good. So here is just a more detailed explanation that DNA polymerase 3, right, once it's initiated, the 3 perform the job. In the end, the DNA polymerase 1, okay, help to remove the RNA primer and then also fill in the gap, right? And polymerase 1, okay, actually polymerase 1, we have 2 fragments, we have clinal fragment, we have 3' to 5' exonuclease activity and the 5' to 3' exonuclease activity, so that they can remove the final RNA primer, okay? Because RNSH can only remove the one in between the 2 RNA primer and RNA sequence, and then the final one has to be removed by the exonuclease activity of the DNA polymerase 1, okay? And the replisome, okay, very important concept, okay? So as we learn, one strand is leading strand, so that means they will just, when they started, they will go all the way to the end very fast, all right? And then the other one is the lagging strand. They have to do the section by section. Then you you will see the Okazaki fragment, one piece here, one piece there, very slowly. However, when they perform the replication, they have to do it at the same speed in order to have both strands to proceed at the same speed. Then we need to have something to hold the leading strand so that they don't go so fast. In the meantime, we also need to put the lagging strand up to the speed to the leading strand. So we have this a setup is called replisome. Okay, replisome is a multi-subunit assembly. Okay, we have DNA polymerase, all right, DNA polymerase, and then we have this the sliding clamp is a sliding clamp, okay, to pull both leading strand and lagging strand together, so that they will make it look like the DNA polymerase 3 to go at the same time. All right, we need to have this the sliding clamp, okay, and also we have the clamp loader, and we call the everything, all right, everything together we call the DNA polymerase 3 holoenzyme, and then it's called replisome. Okay, DNA polymerase 3 holoenzyme, and then it's the replisome. And so basically in this replisome we have two polymerase 3, right, two polymerase 3, and the two polymerase 3, the polymerase 3 has alpha subunit. So alpha subunit is the DNA polymerase activity, and the epsilon subunit now be the proofreading exonuclease, and theta subunit that can stimulate the exonuclease activity. And then we have sliding clamp and then sliding loader so that they can all tie together and to move at the same speed. Okay, so that's called replisome. And then we will finish after this animation. Okay, this is about the replication fork coupling, about the replisome.
During DNA replication, the DNA molecule is duplicated to generate two identical copies. The correct copying of DNA is of vital importance to the cell, as errors that are introduced into the DNA may lead to heritable mutations that could be deleterious to the cell or organism. Thus, elaborate machinery is used both to assure correct copying and to allow the process to be regulated. Coupling of the two DNA polymerases, one for each strand, allows the process of fork movement to be coordinated. To understand how polymerases are coupled at a replication fork, we will first show a standard representation of the replication fork. In this typical representation, two DNA polymerases are moving in opposite directions, with one copying the leading strand of the DNA and the other copying the lagging strand. To expose the single-stranded DNA at the replication fork, a six-membered ring-shaped complex called DNA helicase first separates the parental DNA strands. This separation gives the DNA polymerases the access they need to allow copying. The new DNA strand is always made in the 5' to 3' direction. Because the replication of the two DNA strands is coupled, the leading strand is copied continuously, while the lagging strand is copied in short stretches called Okazaki fragments. Each Okazaki fragment is initiated by a primase that synthesizes the short RNA primer. This primer is later elongated by DNA polymerase. The single-stranded DNA between the newly made Okazaki fragments is bound by single-stranded binding protein, or SSB. In order for the two polymerases to move together with the helicase as the fork moves, the DNA template for the lagging strand must be looped around so that both polymerases are moving in the same direction. With the polymerases now coupled, DNA replication can proceed coordinately on both the leading and the lagging strand. Once the lagging strand polymerase reaches the RNA primer from the upstream Okazaki fragment, the lagging strand loop is released, and Okazaki fragment maturation and ligation by polymerase 1 and DNA ligase occurs. While this maturation is occurring, the primase enzyme that is also traveling with the fork as part of the replisome initiates a new RNA primer on the lagging strand. Once this primer is made, the lagging strand polymerase on the replosome, having completed synthesis of the previous lagging strand, then re-engages with the 3' end of the new primer, forming a new loop on the lagging strand. DNA synthesis thus proceeds with the leading strand polymerase continuously engaged with the DNA, and the lagging strand polymerase engaging and disengaging as each Okazaki fragment is synthesized. To synthesize the lagging strand, the DNA template is looped out while it is coupled to the replosome complex, and the loop is released after each Okazaki fragment is completed. The length of the looped DNA at its maximal size is therefore the length of an Okazaki fragment, which in bacteria is about 1,000 base pairs.
Okay, so that's the ribosome and also the detailed replication. So Ali, yes.
Okay, so just to further clarify, when it loops the lagging strand loops to the leading strand, it stops every single time. The replication does not continue until the replosome is, I would say, like those 2 binding proteins are attached.
Which 2 binding protein? You mean the polymerase?
Yes, the 2 polymerases are attached. Yeah, the clamp is what I'm also
Right. So basically, they will have a slightly opposed. Yeah, not really significant. Oppose them quickly. They move. Yeah.
Okay, so it does. So the lagging strand does. I'm sorry. The leading strand does stop until the lagging strand loops again.
So these lagging strand, they will continuously to come. It's yes, suppose slightly, yeah, not significantly.
Okay, I understand.
In a sense, yes, that's right. You are right. Yeah, they pose a little bit. Yeah. Okay. All right. So yeah.